Ever wonder how your brain talks to your body? Today, we're breaking down nervous tissue and the master map of your nervous system. Let's get started.
This is part 1 of my 3-video series on the nervous system, the brain, and the spinal cord. Let's dive in.
Let's start with the neuron. The soma, or cell body, houses the nucleus, mitochondria, and protein-making Nissl bodies. Structural neurofibrils and receiving dendrites branch outward. Signals start at the axon hillock and initial segment, traveling down the axon, which may have collateral branches. In the peripheral system, Schwann cells create the insulating myelin sheath, leaving gaps called nodes of Ranvier. The outer layer is the neurolema.
When the signal reaches the end, it enters the synapse. Inside the synaptic terminal, mitochondria power the movement of vesicles filled with neurotransmitters and calcium ions. These chemicals cross the synaptic cleft from the presynaptic membrane to the postsynaptic membrane to pass the message along.
Let's dive into histology to microscopically identify neural tissue at various magnifications. First, we have a peripheral nerve in longitudinal section. Notice the 3 connective tissue layers: the outer epineurium, the perineurium, and the internal endoneurium. Looking closer at the nerve fibers, we can identify the axon and its insulating myelin sheath. You can see the Schwann cell nuclei, the outer neurolemma, and the nodes of Ranvier. Finally, observe the structural difference between myelinated and non-myelinated axons.
Neurons are organized into 2 primary categories: structural classification, based on the number of processes extending from the cell body, and functional classification, based on the direction the signal travels. Let's start with structural classification. First, unipolar neurons have a single short process and are primarily found in sensory ganglia. Next, bipolar neurons feature 2 processes, 1 axon and 1 dendrite, and are rare, located in special sense organs like the retina. Finally, multipolar neurons are the most common. They have 3 or more processes and serve as the primary neurons of the central nervous system.
Now, let's look at functional classification, which is based on the direction the nerve impulse travels. First, afferent neurons, also known as sensory neurons, carry information from sensory receptors toward the central nervous system. Second, efferent neurons, or motor neurons, carry instructions away from the CNS to effector organs like muscles and glands. Finally, interneurons, also called association neurons, are located entirely within the CNS. They act as the vital bridge, processing information between sensory and motor pathways. Now, let's identify these structures on a cross-section of the spinal cord. We start at the receptors in the skin, where dendrites pick up a stimulus. This signal travels along the afferent neuron, with its cell body located in the dorsal root ganglion. Inside the spinal cord's gray matter, the signal is passed to the association neuron, or interneuron. From here, the command is sent through the motor neuron, also called the efferent neuron, which exits the cord to reach our effectors, such as muscle cells, skin, or glands, to trigger a response.
Finally, let's classify the neuroglia, the essential support cells for our neurons. In the central nervous system, we find 4 types: oligodendrocytes, which provide myelin insulation; astrocytes, which maintain the blood-brain barrier; microglia, the immune defenders that remove debris; and ependymal cells, which line the ventricles to circulate cerebrospinal fluid. In the peripheral nervous system, we have 2 main types: Schwann cells, which create the myelin sheath for peripheral nerves, and satellite cells, which surround and support neuron cell bodies in the ganglia.
Thanks for watching. I hope you learned something new today. Don't forget to like, subscribe, and join us for part 2, The Brain.